Welcome to Spring LIB.
Hello students, for the given question, first part, to calculate the final concentration of the primer in the PCR reaction, we need to use the formula, that is C1V1 equals C2V2, where C1 is the initial concentration and V1 is the initial volume, C2 is the final concentration and V2 is the final volume. Using the given values, we get C1 equals to 10 micromolar, V1 equals 2.5 microliters, V2 equals 50 microliters. Now plugging in the values, we will get C2 equals C1 into V1 by V2, which will be equals to 10 micromolar into 2.5 microliters divided by 50 microliters, which will be equals to 0.5 micromolars. Therefore, the final concentration of the primer in the PCR reaction is equals to 0.5 micromolar. For the second part, whether the final concentration of primer is within the recommended range of Q5 protocol depends on specific concentration range, the recommended by the manufacturer. Typically, the recommended concentration range for PCR primers is between 0.1 micromolar and 0.5 micromolar. In this case, the final concentration of the primer 0.5 micromolar is within the recommended range. For the third part, Q5 polymerase is a high fidelity DNA polymerase commonly used for PCR. One unit of Q5 polymerase is defined as the amount of enzyme required to incorporate 10 nanomole of DNTPs into DNA in 30 minutes under optimal conditions. The optimal conditions for Q5 polymerase include a reaction temperature of 72 degrees Celsius and a magnesium concentration of 1.5 millimolar. The amount of Q5 polymerase required for 50 microliters reaction depends on the specific concentration recommended by the manufacturer. Typically, the recommended concentration range for Q5 polymerase is between 0.02 units per microliter and 0.05 units per microliter. Assuming a concentration of 0.02 units per microliter, we would need 50 microliters into 0.02 units per microliter, which will be equals to 1 unit of Q5 polymerase. I hope you find the video useful. Thank you.